Hello everybody, my name is MazKhansari and today I'm going to present one of my recent projects which utilizes a 3D nonlinear image registration to quantify medical images. And a specific medical image that was used in this project is optical coherence tomography or OCT images of human eye. Although this technique was tested on OCT, it's a general technique that can be applied to other medical and non-medical images for quantitative computer vision. And I really hope that you find it useful. So here's the outline of the presentation. First, I'll provide a brief background about what is the OCT. So later on, when we see the examples and result, everything makes sense. Then we'll go over the pre-processing and the nonlinear registration technique. And finally, we'll look at the result, how the Jacobian map can be used as a novel visualization tool, and the tensor-based morphometry analysis. To give you a little background about the OCT imaging of the eye, here I'm showing a schematic of human eye. The light gets focused through the back of the eye where the retina is located. And the retina is a multi-cell layer tissue that converts light into electrical signal, and the signal then transformed to the brain through optic nerve head. Once OCT imaging is performed on the retina, the output will show different cell layers, and also on the top we can see vessels that are available on the surface of the retina. In this slide, I'm showing you examples of actual OCT. So on the left side, we can see OCT of a normal subject, and the middle and the right side shows examples for pathological cases that we'll discuss shortly. Looking at the normal image, we notice the different cell layers. They have different reflectivity, and hence they appear with different intensity values in the image. Also, we can see a pit here in the center. This is called foveal pit and is where the light gets focused. So if the shape of the foveal pit is deformed due to disease or aging, then the patient will not be able to see clearly. Each of these volumes that we're going to work on this project comprised of 245 by 245 by 1024 voxels. And the whole volume is made of 2D scans. And these 2D scans are called B-scan in the technical term. And if we look at the central 2D B-scan from this volume, that would be the image on the bottom left. So showing a healthy eye, a clear boundary of cell layers, and a very nice foveal pit. And the disease that we're going to discuss in this project is a complication of diabetes that's called diabetic retinopathy, DR. So this complication happens when somebody has diabetes for a very long time, let's say 20 years, and if they don't treat the disease well, one of the complications that can happen is DR that affects the eye and has multiple stages, starting from mild DR all the way to proliferative diabetic retinopathy, which can result in vision loss if it's not treated in a timely manner. So looking at the mild diabetic retinopathy case, we can see some shape changes in the volumetric view. And if you look at the central 2D B scan, we can see fluid packets that are technically called edema, and we can see changes in thickness of some layers that are sign of the disease. And if you look at the most advanced stage of the disease, in a volumetric view, we'll see the entire tissue is expanded and the foveal pit is completely missing, which is a sign that these patients will most likely not able to see. If looking at the 2D B scan, we can see massive fluid packets areas. We can see hard exudate, these bright areas, also a complication of DR. Some layers are missing. And the goal of this project is to propose a novel 3D nonlinear registration technique to quantify the extent of these changes in the R patients as compared to a normal control group. So the project includes some pre-processing step. As you can see in the original OCT, there's a lot of speckle noise that we would like to remove. So I used non-local means denoising, which results in the denoised image on the top right. You can see the noise has been removed, and at the same time, layer boundaries are very intact and detail of the image is not affected. Another pre-processing step is to mask out the background because it will not contribute to a registration task. And also, in this specific OCT case, there's a layer on the bottom of the tissue. It's called choroidal layer and comprised of small vessels called capillaries that are highly variant between the subjects. So we cannot reliably register this part of the image, and it's better to remove it to improve quality of the registration. So at the end, the pre-processed image that goes through the registration pipeline is shown on the bottom right. It's important to note that these pre-processing steps and the rest of the steps are all performed in a volumetric level. However, to provide a better visualization, in this and the following slides, I'm only showing you a 2D B scan that's exported from the middle of the volume. So talking about image registration is a task in which we would like to find a one-on-one correspondence between two images. Let's say if we have image 1 on the left and image 2 on the right side, we want to find a transformation to map each pixel, let's say P, to a corresponding pixel Q in the second image. And we would like to do it for every pixel in the first image to the second image. So overall, the whole problem can be formulated as an optimization problem in which we'd like to minimize the cost function C to find the transformation for finding one-on-one correspondence between the fixed and a moving image. To do reliable image registration, we need features that can be correspond between a moving and a fixed image. Looking at this specific example, we can see the layer boundaries are a good feature. The top and bottom of the retina is a reliable feature, and also we have foveal pit. If we look at the volumetric view, we can see the foveal pit on the center, and if we cut through the volume through z-axis, we would get the image on the right side showing foveal pit's appearance in a 2D view. So this part of features, understanding the features for registration is application dependent. If you are going to register different types of images, you first need to make sure that there is enough features and these features are corresponding between fixed and moving image. So now we know about image registration and the features in OCT, we go ahead and apply two-step image registration. First, I'm applying an affine registration to bring the two volumes as close to each other as possible. And note that affine is a rigid transformation. There's no nonlinearity here. The goal is to bring the two volumes as close as possible so that in the next step, we can apply a nonlinear registration to match details of the two tissue. And the whole registration pipeline was performed in Elastic software. Here I'm showing the example of a moving image. And the goal is to apply an affine transformation to match it with the fixed OCT. So the goal here would be to correct the orientation and also the location. So we'd like the foveal pit shifted slightly to the right to match with the foveal pit of the fixed volume. And also we want to correct the scaling. If one is thicker than the other, we would like to correct that. One important consideration for this specific application is that if we define axis of z, x, and y as shown on the bottom right here, we'll see that along the z-axis, we have different cell layers with different intensity values, layer boundaries that can guide the registration. So if the affine wants to do stretching along z-axis, it has enough features. However, if we look along x or y-axis, the only feature is the foveal pit, which is generally located on the center of the image. So if you look close to if you're in the border of the image, there's not enough features to guide the stretching of affine. So in this specific example, let's say if we apply affine registration to register this moving image to the fixed image, because we want to move the foveal pit slightly to the right, and also we correct the orientation, we expect to see some blank areas on the left corners of the image. So this image, the right bottom image, shows a good, reliable affine registration that, as expected, showing blank areas on the left border. However, if we apply a vanilla affine registration, due to lack of features on the left border, it will stretch the tissue to fill out the image. So to avoid this, I applied a modified version of the affine, and I used with restriction on stretching along x and y axis. So once we apply the affine registration, we define an area of interest in which the fixed and moving image are close to each other, and then we go ahead and apply nonlinear registration to match detail of the two tissue. So in this slide, I'm showing you some detail of nonlinear registration. So B-spline transformation was used with a normalized mutual information cost function, and the optimization was achieved by stochastic gradient descent. In the affine phase, 8 intensity pyramid resolutions were used. This is because these are images of the eye, and the patient may not be able to fixate very well during the image acquisition phase. So the tissue may appear everywhere in the image, might be far on the top or the bottom, might be rotated extremely. So we would need more intensity pyramid resolutions to step by step bring the tissue in the moving image to that of the fixed image. For the nonlinear part, the 4 resolution was enough. At each resolutions, the number of samples and number of iterations are shown here. And finally, you can see the equation for normalized mutual information, which uses a Parsons window that is defined here on the bottom. And I'm not going to deep dive into the mathematics here, but the NMI mathematics is well established, and I encourage you to study the papers, and also I would be happy to answer if there is any question about this or other part of the presentation. So once the registration and pre-processing pipeline is established, we perform the registration, and here I'm going to show you some example result that we can get by this technique. So for a couple of following next slides, always the moving image is shown on the top left, the fixed image would be on top right. Bottom left shows a affine registration result, and bottom right will show the nonlinear result. So here we have a mild case of DR. We can see some thickness changes. The foveal pit is very small. Also some other thickness changes on the left side of the image. So once we finally register this moving image to the fixed image, we get the affine registration result on the bottom left. So here we notice the orientation is corrected. The foveal pit is slightly shifted to match with the foveal pit center of the fixed OCT. And also the tissue is stretched along the z-axis to get the thickness of the fixed volume. So now the finely registered volume is close enough to the fixed OCT. Then the nonlinear registration is performed to match the detail of the anatomy. And the bottom right image shows a perfect nonlinear registration result. Another example here, more severe case of the disease with large fluid packs areas to show you how these are deformed after the registration. a fine registration result after moving to the fixed volume shows the correction of the orientation, location, scaling, and also shear. And the final nonlinear registration shows how the moving image takes the shape of the fixed image. And later on, we'll see that we use the amount of deformation from the moving to the fixed OCT to quantify and detect different abnormalities in OCT. And also it's important to notice that these fluid areas are shrunken here, but I'm regularizing the registration task so it does not shrunk everything and smooth out every detail of the image. Another example of a more severe case with huge fluid areas everywhere in the OCT, lots of some layers, a result of a fine registration, and finally the nonlinear registration shows how the huge fluid areas are shrunken, so the OCT takes the shape of the fixed one. And finally, another result of an advanced stage of the disease, in which case, instead of the fluid packets that make the tissue expand, here we're facing with a case of loss in the tissue. So we can see the foveal shape is completely changed, and also these bright areas called hard exodus is visible in this 2D scan. Once we do the fine registration, the tissue is expanded to get the thickness of the fixed OCT. And finally, the nonlinear registration will match detail of the moving image with the fixed OCT. So once we set up the registration pipeline, we need to validate accuracy of the registration. To do that, I used two different matrices. First was dissimilarity. That's used to calculate the overlap of two regions, and it's equal to 1 for a complete overlap case. And to calculate DICE, the retinal tissue was divided into 3 different subregions. They are called superficial, deep, and avascular. And this category is based on type of the vessel that appear in each region. So the superficial includes the small vessels, deep layer has capillaries, and anything beneath that up to this red line on the bottom is called avascular because there's no vessel in that region. And this is a common classification of retinal part by clinicians. So in 5 normal and 5 diabetic retinopathy case, the volume was registered to a fixed normal volume, and the amount of overlap for each of these layers after the registration with that of the fixed OCT was quantified by means of dissimilarity, and the result for the normal subjects are shown on the top table. The result for DR subjects are shown on the bottom table. So we can see very high dissimilarity for all the 3 regions. The deep layer has slightly lower DICE compared to the other 2 layers, which can be because of its smaller size. And the values for the DR subjects are slightly lower than normal and healthy cases, but there's still very good dissimilarity, very high values. And if we combine all 3 layers together, the dissimilarity would be almost 1. Another metric that was used for quantifying accuracy of the registration was average symmetric surface distance between each of these 3 categories with respect to the fixed image after registration. And here we would like to see small values, and the values are in micrometer. So we can see that the average symmetric surface distance is pretty small for each of these 3 categories, and also very small when we consider the entire tissue. Same result for the DR cases. Also, please note that for simplicity, here I'm showing the result only based on these 3 categories, but in the actual published paper, which I will show you in the coming slide, we could, I categorized the retina into 8 different cell layers, because clinically speaking, there are different cell layers within each of these subcategories. And I also reported dye similarity and average symmetric surface distance for each of those layers to show how accurate and perfect this method can align each of these sublayers. Also, we can compare the result of the result of registration with layer segmentation. So I put this slide here because generally OCT images are quantified by means of layer segmentation. So the layer segmentation methods generally use a graph cut algorithm or recently emerged of deep learning methods. The purpose is to segment each of the retinal layers and finally calculate the thickness, overall thickness in each layer, and use that as a metric to compare the thicknesses in normal or disease, and also to help with diagnosis different eye diseases. So if we look at this example on the top left, the subject have some complications. We can see hard exodus and small fluid areas here. Once we apply an open source, widely used layer segmentation tool, we see that it misses some of the layers that can dramatically affect the result. But if we apply my proposed technique of nonlinear registration to register this moving image to the fixed image, we can see that these fluid packets and also the hard exodus are well taken care of, and the moving image is perfectly showing, taking the shape of the fixed image. So this can be advantage of using this technique over only layer segmentation for quantifying the OCT images. So in this slide, I'm trying to show you the process of generating the atlas for registration and what benefit we can get from it. As you remember in previous slides, I mentioned that we always need a fixed image. So that would be the target, and a moving image to register to the fixed image. So generally speaking, we could use one individual image as the fixed and then register rest of the images to the fixed image. Or we could generate an atlas, and there are different ways to generate the atlas, and I'll explain the one that I applied here. So if we use the only one fixed OCT, we can see this could be the reference for all other moving images. We can find a good-looking OCT that's not tilted and centered, the fovea is centered, and use that as the reference. This would be the volume for the reference. And if we look into a cut through the OCT, we can see that in the superficial layer there are vessels, and which may not necessarily correspond with the vessels of the moving image. So to further improve the registration result, I generated an atlas based on this fixed image by registering a group of healthy and normal looking OCTs, which includes 24 for this project. So I nonlinearly registered each of them to the fixed OCT and then averaged the result and repeated this process for 5 iterations to get this smooth atlas on the bottom left. The volumetric view of the atlas is shown on the middle, and also if we look at the cut through the atlas, we see due to the smoothing process, all the vessels are gone. So this can dramatically improve quality of registration in the superficial layers because the vessels do not correspond from one OCT to another, and this can be misleading for the registration. So generally, when we use atlas, we're reducing the bias by smoothing intensity values, as we can see here when we compare it to the individual image. But also, there is a risk that if the base OCT that was used to generate the atlas, if it's not representative of the normal group, then we'll have extra bias in the model. An alternative is to use group-wise image registration and generate an atlas by using all images in the normal group. So once we have the atlas and the registration pipeline, we go ahead and register all the images that we have in this project, the mild and moderate and severe diabetic retinopathy, and also the proliferative diabetic retinopathy cases. And one result we can get from it is the Jacobian map by calculating the partial derivative of the deformation field for each registration case with respect to the atlas space. So the Jacobian map reflects how values of the atlas are supposed to change to take the form of the moving image. For example, if this is a moving image that we would like to register to the atlas, the Jacobian map is shown here that shows some values of the atlas are supposed to shrink and some values might have to expand to match with that of the moving image. So the color map shown on the Jacobian map is shown on the right side here. The values are quantitative. 1 means no change, and let's say 1.3 shows 30% expansion, 1.5, 50% expansion. Similarly, 0.7 shows 30% shrinkage of the atlas to take the shape of the moving image. And these can serve as a novel visualization tool for clinicians to look at the tissue and get an intuitive understanding of what is happening to different parts of the OCT due to disease progression. For the same example, if we look at the cut through the center of the moving image and a cut through the center of the atlas, we notice that the atlas is supposed to expand on the central foveal part to take the shape of the moving image. And this is clearly reflected as expansion values in the Jacobian map. Another example is shown on the right side. So we can see some expansions are happening on top of the fovea that are reflected in the Jacobian map. And also a cut through the volume can show clearly how the big expansion is needed for the central part if the atlas wants to take shape of the moving OCT, and that is correctly reflected in the Jacobian map. Here are 2 other examples, and these are the same subjects and same OCT for which I showed the B-scan result of affine and nonlinear registration earlier in this presentation. So a proliferative stage of disease requires huge expansions in different retinal areas that can be also visualized when we cut through the volume. And an interesting property of Jacobian map is that we can see local changes due to the disease. For example, in this case, we can see many expansion areas. However, there are some dark blues here on the right side showing that this area is shrinking due to disease progression. And if we look at the final example that was related to a subject that lost part of the tissue, we can see this loss is perfectly reflected in the Jacobian map. And since the Jacobian map values are quantitative, we can go ahead and perform group-wise statistical tests. So for this purpose, I'm presenting a tensor-based morphometry approach in which we would like to detect voxels that are statistically significantly different between different group of subjects. So we have a reference group of NC representing normal control with 24 OCT image volumes, and 2 different groups. The first one would be NPDR, which is the non-proliferative stage of diabetic retinopathy. And the way the statistical test is carried out is that if we consider one voxel, let's say here, we would have the Jacobian map values for this voxel for all these 24 normal cases, and also the same Jacobian map values for all the 57 NPDR cases. And we can do a voxel level statistical test to see if these two vectors are statistically different from each other. So the test was, I used a t-test, a one-sided t-test to test separately for shrinkage and expansion, and the result of statistical significant shrink areas are shown by blue, and the result of significant expansion areas in the NPDR group compared to normal group is shown by red on the right image. Also, the TBM, I applied it to the proliferative stage of the disease, which is the most advanced stage of the disease, and we can see the area of significantly shrunken and expanded are higher compared to NPDR. And also we can see many significantly expanded areas are around the fovea, which matches with the current clinical knowledge. So overall, the TBM presents a method to do group-wise statistical analysis, which can help with identifying new biomarkers and better understanding the progression of the disease. So this study was published recently in the Journal of IEEE Transaction on Medical Imaging, which is the probably the most prestigious journal in the field, and I would encourage you to take a look at the paper and reach out to me if you have any question. So in summary, a novel technique for 3D nonlinear registration of OCTI images is presented. The method is validated based on location of the fovea and different cell layers, and there a method for generating atlas for normal subject is presented, which can reduce the bias for the registration. The Jacobian map is presented as a novel visualization tool that can help clinicians to better understand individual cases. And finally, tensor-based morphometry is performed for group-wise voxel level statistical analysis. So my name is MazKhansari, and I have a PhD in biomedical engineering and a master in electrical and computer engineering. The focus of my work is on computer vision, data analysis, and machine learning. Thank you very much for listening